So in Unit 3, we just have two chapters. We have Chapter 11 and then we have Chapter 12. And so in Chapter 11, now that we have an understanding of what microbes are from Unit 1 and we have an understanding of what they need from Unit 2 and the processes they go through to gain those things they need, now in Chapter 11 we can start looking at how to control microbes. And so two basic control methods we're going to look at in this chapter. We're going to look at how to control them physically and we're going to look at how to control them chemically. And so to start off, very first objective, the three major categories of control will be physical, chemical, and mechanical. So these are the three ways that we seek to either destroy a microbe or to reduce the microbe in a given area. And so as far as these are concerned, just to give you kind of a quick rundown, so the physical agents of control, these are going to be conditions that you can alter. So physical conditions around them. So heat is one of them, whether it be dry heat or moist heat. We can also use radiation as far as a physical agent, ionizing or non-ionizing. When we look at mechanical, this is ways that we can physically or we can mechanically remove them. So we look at filtration methods like filtering air, filtering liquids, and then of course we have the chemical means of control, which would be adding things to them. So whether these be gases, whether these be liquids, of ways that we kind of control them. And so as far as microbes are concerned, these are our primary targets. So we're targeting vegetative bacterial cells and endospores. We're targeting fungal hyphae. We're targeting fungal spores, yeast. We're targeting protozoans, whether they be in their trophozoite or their cyst stage. We're targeting those helminths. We could be targeting viruses, and then of course we could target prions. And so one of the things that we know about microbes is that microbes have levels of resistance. And so three categories we look at as far as resistance is concerned. So we have those that have the highest resistance to control, moderate resistance, and then the least resistant. And so if you look at the words highest resistant, when we say highest resistant, these mean these are the hardest ones to kill. So these are the ones that are very resistant to control methods, whether they be physical, chemical, or mechanical. They're just really hard to deal with. And so two basic microbes that we see in this category, one are the prions, and then two are the bacteria that have the ability to form endospores. And so these are typically the hardest ones to deal with. And if you think back to previous units, for prions, we really don't have anything that we can use to remotely deal with them. And then endospores are just really, really hard to deal with because they're able to withstand those unfavorable conditions. So then you have moderate resistance. This is going to be where a lot of the microbes we look at protozoan cysts, the dormant stage are here. Naked viruses are moderate resistant. Bacteria that don't have endospores but have resistant cell walls, so like Mycobacterium, Pseudomonas. A lot of the staph have moderate resistance. And then we have the least resistant. Least resistant, one of the key things you'll want to remember with this category are the bacterial vegetative cells. Now, that vegetative there, you got to be careful with it because a lot of times when we use that word vegetative, you think it's something that's not moving. Like if we think of a human that's in a vegetative state, they're in a very dormant state. But for microbes, when we call them vegetative, those are the states that are actively growing. They're actively reproducing. They're actively participating in chemical reactions. And so most bacterial vegetative cells, those very active cells, are least resistant, which means they're the easiest ones to actually get rid of. So when we look at controlling microbes, we're going to talk through some different control methods. And these control methods are going to look at two basic sides. So you have what's called cidal, and then you have what's known as static. Now, these suffixes are actually at the very end of objective number 3, but let's just go ahead and look at the first one, cidal. Anything that's referred to as side or cidal, it means to kill, right? So you think about homicide, you kill a human, right? Bactericide, it's going to kill bacteria. Fungicide, it's going to kill fungus. Sporicidal, kill spores. Now remember, viricide, you can't really kill a virus, but it's going to put them inactive. And so if you look underneath it, these are three different methods that we can use to kill microbes. So the first one's called sterilization. And the key thing to know about sterilization is if you use a sterilizing technique, the goal is to destroy all microbes. So what sterilization does is it makes it free of everything. So endospores, you can get off of it. There is the potential maybe that prions could be taken off of it as well, because again, we're destroying all of them. Now you have to remember with prions, there's not a lot of knowledge about them, so it's not something that they're very rare, so we don't see them very often. We don't know that much about them. But with endospores, we know a lot more, so we can tell you that sterilization procedures will totally wipe them out. If we look at disinfection, so the key thing with disinfection is it's a physical process or it's a chemical process, and the goal here is to get rid of most. So when you disinfect an area, you're not going to wipe out all of the microbes, but you're going to get rid of most of them. The things that linger typically are going to be those highest resistance, so those endospores that we talked about earlier. And then antisepsis here is a third way to kill stuff. The key thing to note about antisepsis that makes it different from disinfection is that typically this one's going to be used to body surfaces. Okay, so disinfection, we typically associate it with inanimate objects, where antisepsis, we associate that with body surfaces. Again, what they have in common, though, is that both disinfection and antisepsis are going to wipe out most everything. So if we look at just some quick examples, so when I think of sterilization, I think of what we do to a lot of equipment. Right? We sterilize equipment, whether they be utensils that are used in a dental office or in surgery. Those materials are sterilized. We want them free of all. Disinfection, that's what we do when we wipe down the countertops, right? So when you clean the surfaces of your kitchen, when you clean your toilet, you're looking to disinfect them. Antisepsis, as far as body surfaces, I'm thinking hand washing, right? Or example they give on the screen where you're trying to kind of disinfect wounds or surgical incisions, so you would use antisepsis to do that. So then if it's not cidal, we categorize it as being static. So what static methods do is they attempt to slow down. So they attempt to kind of stunt or prevent their growth, prevent them from multiplying, but they don't necessarily kill them. And so a couple of words that are mentioned under this one, they mention sanitation, they mention degermation. So sanitation, cleansing technique, it removes microbes from inanimate surfaces, and the goal here is to reduce the potential for infection, reduce the potential for spoilage. So when you sanitize something, we sanitize maybe foods or beverages, things like that, but again, we know that it's not getting rid of everything, and what it's mainly going to do is to kind of slow it down. They also mentioned degermation. So again, you're limiting the amount of bacteria that's on it, typically by some type of mechanical means, but again, noting that you're not killing anything, you're just trying to keep it from spreading. So here's a great chart that you can kind of look at. It's a table that it's found in the book. It's table 11.2. So if you're kind of looking around, this is on page 332. It goes back over what these major terms mean, and then some really good examples of how you would use them just in everyday life or in a healthcare setting. So your fourth objective says, okay, so the goal here for most of them is to kill them, right? Cidal, we want to kill the bacteria, we want to kill the microbe. And so what does that look like for a microbe? Because for a human, we know what death looks like, right? We know it's absence of brain waves, absence of a heartbeat, those types of things. But when we think about microbes, they don't have brain. Right? They don't have a heart. They don't have those types of tissues and things like that we see, and organs that we see in humans and other animals. And so it's very hard to detect if a microbe is alive or grows. So what we look for with microbial death is we look for them to lose the ability to reproduce. So if they lose the ability to reproduce, we consider them being dead because they're no longer viable for growth, no longer viable to actually spread. So that's what we're looking for with those. Now, as far as some factors that can affect their growth, so the number of microbes can affect, as far as it can affect their death. The nature of the microbes in the population can affect their death. So we think about how many are actually there. So if you have an overabundance of microbes in an area, that's going to cause some of them to die off, right? The environment can be a factor that affects death. So temperature, pH. Remember, we learned in chapter 7 that a lot of microbes have specific temperature ranges that they like to be found out, or they have that specific 6 to 8 pH. So if you put them in an adverse temperature or an adverse pH, you can kill them. Whatever agent you're applying to them, so the concentration or the dosage of that agent, that chemical that you're adding, can affect their death. And then of course they mentioned the presence of water, the presence of organic matter, and other types of inhibitors can obviously affect their death. So one of the things we want to hit on here, and you're going to hear this come back in this chapter and in chapter 12, is that as far as microbes are concerned, there's basically 4 cellular targets. So 4 parts of these microbes that we target with antimicrobial control. And so targeting them physically or chemically in this chapter. And then when we look in chapter 12, we're going to look at the targets of antibiotics and things like that. But as far as physical and chemical control is concerned, 4 parts of the cell that we target. This is objective number 5. So the first thing we can target is we can target the cell wall. Now remember, most microbes have them, right? So fungi have cell wall, bacteria for the most part, except for those couple of exceptions, they have a cell wall. And so when we look, algae have a cell wall, right? Remember, the only ones that really don't have a cell wall we looked at were like protozoans, helminths, things like that. So as in targeting the cell wall, it's a common part of a cell that a lot of our microbes have, and what we typically try to do with it in targeting it is we try to make it fragile. We try to make it weak, because if we can weaken the cell wall, remember that the function of the cell wall is to protect it, right? And so if we can weaken the cell wall, we can cause the cell to lyse. So we can cause the cell to take on liquids, and we can cause it to burst. So cell wall being the first target that we have. The second thing that we can target is we can target the cell membrane. Now remember, the benefit of this is all cells have one, right? So regardless of what type of microbe they all have a cell membrane, and so it's always a common target that we can have. And the ability here, remember, the purpose of it is cell membranes are typically selectively permeable. So if you can target the cell membrane and you can compromise it, you can cause it to lose its integrity. So you can cause it to lose its feature as being a barrier. You can cause it to lose some of its selective permeability, and so then you can allow materials to go into the cell that would normally be kept out, and in turn you can damage or kill that cell. A third thing that we target is we can target proteins, we can target nucleic acid synthesis. So in order to target proteins, in order to target the building and the replicating of DNA and RNA, we typically will prevent some processes. So we can prevent replication, which is what makes the nucleic acids. We can target transcription, translation, and we're talking about the fact that those are the 2 events in protein synthesis. We can target the bonding, the peptide bonding of amino acids that actually build proteins. And so in doing that, if you remember, the benefit of that is proteins basically do everything, right? So proteins are carrying out all of these chemical reactions that are happening in cells. So if you can target proteins, you can shut those chemical reactions down. And in targeting nucleic acids, remember, nucleic acids are needed to make the proteins. So again, you would affect proteins with this one. But another thing that you can target is if the proteins are actually able to be made, you are actually able to target the function of those proteins once they're made. Remember, we talk about this in biology, shape determines function. So as long as the protein is the appropriate shape, it's going to do its job. So one thing that we can actually do is we can target the shape of a protein. We can denature the protein, which means we make it lose its shape, and if it loses its shape, in turn, it loses its function. So 4 targets that we have, very important to remember as we move on. And so just a quick check here for you in going through this.